All right, now we're going to do agents of evolutionary change. And don't worry if you're more interested in those homologous structures, the vestigial structures, the fossils. We're going to go back to that. But right now, what I want to do is I want to kind of connect it more to genetics, since that's what we just finished, and this like more microevolution. So dealing with the smaller scale changes and talking really about that natural selection, because that's going to be one of the keys for this unit. All right, so today what we're going to talk about is agents of evolutionary change, or in this video. You should be watching two videos today. The agents of evolutionary change. So we've got 5 main things we're going to talk about. So one is mutation. Now, mutation is any change in the DNA sequence. Now, what I wanted to remind you is that when we have a change in the DNA sequence, that doesn't always change the amino acid sequence. It doesn't always change the protein. Even if it does, that doesn't always change the structure or the function, but it can. So if we have a change in the DNA sequence that then does change the amino acid sequence, remember that's going to be more likely if it happens during the first and second amino acids in our codon, because remember our codon would be something like this. And this last one can often be a swing. So if you look at your codon chart, a lot of times you might have, and this is a bad example because this would be really a UG, and that would be the start codon. But a lot of times what we have is we have things like, let's do CCC. CCC is probably the same as CCG, CCA, and CCU. Usually we have this last one will swing, and at least 2 of them will be the exact same amino acid. And I'm not sure if that's correct. I can't look it up on a codon chart right this second to make sure which amino acids are coming from this codon, but we will usually see that we don't get a change in amino acid if we're changing that last nucleotide in the triplet. If we get an amino acid change, that might change the protein. It could change it dramatically. Remember, sometimes if we change to a stop codon, that can change the protein by making you have only half a protein. If we change it from one amino acid to another, that might change the structure. It depends on what amino acid is changing. So if we're going from a like acidic amino acid to a basic amino acid, that will definitely change our structure. And a lot of times structure equals function. If we change our structure, we're going to change the function. Now, if all of those things change, we will often see this change in phenotype and therefore fitness. But again, there's question marks at each step because it depends on where that mutation is, whether each thing is going to happen. All right, next I want to talk about gene flow. Gene flow is a concept where we talk about movement of individuals into and out of populations, and that this can cause the population's genes to change. So when we're looking at a population level, because remember, evolution does not happen in an individual, evolution happens in a population. Now, this can happen a lot with things like seed and pollen distribution, because seeds and pollen are going to be distributed basically randomly, especially seeds that are distributed by wind. Animals can migrate. So you might have subpopulations that have very different allele frequencies. So for example, this subpopulation has all recessive alleles. This subpopulation has all dominant alleles. If one of these birds goes over to this population, this causes this genetic mixing. This adds in new alleles into this more dominant population. It reduces differences. So if we have gene flow, it reduces differences between 2 populations and makes them more similar. So gene flow is the exchange of genes between 2 populations. Next is non-random mating, or mate choice. So I put 2 pictures here of examples. Birds are the best example. We almost always use birds as examples in biology because they display their mate choice very vibrantly. The males tend to be bright colored, the females tend to be more drab, and then they can be choosy. So this example with the peacock. The peacock has all these bright feathers, the male does anyway, and the female is more like brown, and it can pick what male peacock it wants to mate with. Cardinals, which I've been seeing out and about lately, the more red the cardinal, the more desirable he is from a mating standpoint. One of my friends in grad school did a study on it, which she called having good genes. Good in quotes there, that, the red cardinals seemed to have the better genes because they were sought after by the female cardinals. This is an example that you may not have seen too much before. This is an example of an aquatic creature that makes these patterns in the sand. And it makes these patterns in the sand in order to attract a mate. So it's almost like sand art. So these fish, trying to remember what kind of fish is it. It's the puffer fish. That's what I thought. The puffer fish. I just had to Google to double check. So the puffer fish will actually make these patterns in the sand as part of the mating ritual in order to attract the female. So that is another example of non-random mating. So the female is choosing a mate based on this behavior, which means that her, puffer fish pups will be more likely to exhibit that same behavior. Genetic drift is another example. This happens due to random sampling. We have two examples that we like to talk about, the founder effect or the bottleneck. A founder effect is when we have a small group that starts a new colony. So for example, we have a red bird or a couple red birds that fly to this island. They start a new colony. Because this colony was started by only red birds, it doesn't have those yellow alleles. The other one is a bottleneck. And in a bottleneck, let's get this out of the way. In a bottleneck, you have some factor that comes through and reduces the population to a small number, and then the population expands again. But the key is that it is random. Oops. So the key here is randomness. So we don't have something that comes in and removes all the red. It's randomly coming through and just taking a whole bunch. So if you do that, if that bottleneck happens, you see we went from 20% of each color down to we still have 20% green, we still have 20% red, but we only have 0.14 pink, 0.06 yellow, and 0.4 blue. So by this random thing, we have changed our frequencies. And this is, again, removing numbers until we get to this 10th generation. And in this case, we're just removing individuals to see how we can change our percents. So by this time, we have 0.2 blue, so we're back to 0.2 for blue. But yellow and pink have both gone extinct, and red has gotten to be the most in the population. And this is all randomness. Here's another example of that bottleneck. So this is why we call it a bottleneck. You have this parent population. Imagine it's in a bottle and you're pouring it out, and only a couple can get through the neck of the bottle at a time. So there's some disaster. It means that most of what's in that bottle will not survive to the next generation. And so some of the alleles are lost from the gene pool, but it's not due to fitness, it's due to randomness. So this red allele is just not very common, and it's completely lost to the next generation. Also, the yellow is about as common as the blue, but as you can see here, because of just the randomness of the bottleneck, we're left with mostly blue individuals. So this narrows our gene pool. And remember, that is part of your genetic drift. Cheetahs are a good example. All cheetahs share a small number of alleles, less than 1% diversity. It's as if all cheetahs are basically identical twins. We think this is because of two separate bottlenecks, 10,000 years ago at the end of the Ice Age, and then the last about 100 years ago due to poaching and loss of habitat. So based on their genetic information, based on looking at different mutations, we're pretty sure about when those two bottlenecks would have occurred. And then last is natural selection. We've talked about natural selection a lot. The less fit animals are removed, in this case, eaten by this bird who was hungry. It can see the blue caterpillar more easily on the green leaves than the green caterpillar, and so it can eat those. You can also have differential survival due to environmental conditions. So things like climate change, food source availability, predators, parasites, diseases, all of those things can cause differential survival. Based on the genetic makeup of the individuals. And then also toxins. So like toxins present in food items or toxins present in the environment in other ways. So like this could be people adding them. The combination of alleles that provides fitness will increase in the population, and then you will be left with those fit individuals. All right, so for practice, give an example of genetic drift that is not an example from the video. So make sure you're thinking of some other example. It does not have to be one that is real. You can make something up random. All right, that's it.